Research is a lot. There are tons of procedures, experiments, and trial and error. Sometimes things work, and sometimes they really don't. It's a learning curve to say the least. So, here's what my summer in research looked like. But since the Zuckerman has a strict no photography policy, I'll show you through animation instead. Behavior. I started with behavioral testing, specifically the classic tube test. It's exactly what it sounds like: a tube, a camera, and two mice. Tube tests help establish social hierarchy. To explain that, let's start with a human example. This summer, I worked with Erica, my boss, and Caleb, my lab mate. Obviously, Erica and I aren't at the same social rank. She's at the top of our lab's pyramid, while Caleb and I are at the bottom as undergrads. Humans establish hierarchy through things like job title, education, age, and other social cues, many of which involve vision. Mice do something similar, but they rely heavily on olfaction. In the tube test, two mice compete until one pushes the other out. After 4 consecutive days of consistent results, we can then establish their social rank. The winner is rank 1, and the most submissive is rank 4. But hey, at least the rank 4 mouse is winning something. Then, after establishing their hierarchy, I presented the mice with urine. Why urine? For mice, urine isn't just pee, it's social information. It contains chemical cues about things like identity, sex, and social status. So when a mouse smells another mouse's urine, its brain is processing socially meaningful information. And that's where my project comes in. We're studying the ACC and BLA brain regions involved in social and emotional processing, and specifically, oxytocin receptor expressing neurons in the ACC. And oxytocin isn't just the love hormone. It can help regulate social salience, which means how important social information is to the brain. So we're asking, how does the circuit respond to socially meaningful odors, and what role might oxytocin play? To understand that, we'll have to look inside the brain. Unfortunately, that means euthanizing the mice, but we got their brains. Histology. Next came histology, which means preparing the brain so that we can see what happens at the cellular level. I sliced the brains, used immunohistochemistry to label specific proteins, and imaged the tissue under the microscope. And honestly, the fluorescence is gorgeous. But eventually, those pretty images become data. Analysis. Right now, I'm using an AI-based cell counter to quantify oxytocin receptor expressing cells in the ACC, and I'm quality checking the results. Once that's finished, I'll be able to statistically analyze the data and see what we found. Reflection. And that's my biggest takeaway. Research is a lot. It's detailed, time-consuming, and full of trial and error. But I've learned how to troubleshoot, problem solve, and adapt. Science isn't easy, but maybe that's what makes discovery so rewarding. I'll be continuing this project in the fall, and I can't wait to see what we discover. Thank you to the CCE Summer Funding Program and its donors for making this experience possible. Your support gave me the chance to focus on the science, discover what I love, and worry a little less about everything else. I'm all out.